Homeostasis is a control pathway in the body. To understand it, we'll use a graph. So during homeostasis, your body controls different variables in a set point. Make that set point right here. In between that set point, there's a normal range. In order for your body to control things, you have different variables that move you toward or away from your set point. So homeostasis is whenever your body controls anything inside of this normal range. There are different examples of homeostasis in your body. One of those examples is sweating. When your body starts to overheat, you will start to rise in temperature, and as you move away from your set point, your body will tell your sweat glands to open its pores and to start sweating to cool down your body. As your body cools, it brings you back towards that set point. In your body, you have two different kinds of feedback that comes along with homeostasis. The first is a negative feedback loop. That negative feedback loop is anything that brings you away from the normal range, or away from the set point, back toward the set point. In that previous example of sweating, that is a negative feedback loop because your body has overheated and moved away from set point, and then it started to sweat, which brought you back toward the set point. An example of a negative feedback loop is something that moves you away from the set point and then does not return. It would move in this direction away from the set point and then out of the normal range. Anything outside of this normal range would require medical intervention to help you bring that into a negative feedback loop and start moving back towards the set point. An example of the positive feedback loop is blood clotting. When you have a cut, your blood is moved toward that cut so that the platelets that start to enter that wound release chemicals that attract other platelets to come. So those red blood cells are moving away from where they should be and go to help that to help return your body to homeostasis. When that blood clotting has completed and your body starting to heal, it's in that negative feedback, returning back to its set point or its normal range. This is all controlled by your reflex control pathway. In your reflex control pathway, you have stimulus that occur in your body, and the stimulus will affect a receptor somewhere in your body, and anything that moves from a receptor to a control center, this is known as an afferent pathway. So there's the stimulus affects the receptor, and the receptor moves toward the control center. That is the afferent pathway. The control center is often just the brain. It can be anything in the central nervous system, but it's usually just known as the brain. Anything that moves from the control center to an effector is known as an efferent pathway. And those efferent pathways move into the effectors, and that causes something in your body to be altered back to your receptor. The variable will be changed, and that will help you maintain homeostasis. There are a few other types of homeostasis. The feed-forward loop is a type that will give you anticipatory response. Common examples of that are when you are in the presence of sight or smell of sour foods, then your mouth can water, your salivary glands will start to excite. And then you also have redundancy and acclimatization homeostasis. When your body has something vital it needs to take care of, sometimes it will build in systems that if one fails, another will kick in and maintain that same state of homeostasis. And acclimatization homeostasis is when your body will take effect into changing something over time to create a new state of homeostasis. Common examples of that are when you move to higher altitudes, it takes your body time to adjust and your red blood cells to adjust to the higher altitude and a difference in oxygen. But over time, you create a new state of homeostasis through acclimatization. Particles can pass through the extracellular fluid to the intracellular fluid through what is known as a plasma membrane. Those plasma membranes are made up of lipid bilayers, which I've drawn here. But they diffuse particles through channels. When they have these facilitated diffusion through channels, for all 4 of the types, they do not require energy of any kind. It's all just facilitated by these channels that are here. So the first channel that we'll talk about is a leak channel. That's this right here. The leak channel is always open. It can flow in either direction depending on where the gradient is, and the gradient always flows from high to low. These leak channels can be, one example can be of potassium. So we can have potassium moving through this channel. I'll call this the ECF and this the ICF. So it's moving from outside to in with no extra effort from energy. It's just an open channel. There's no gates on it. It is specific for each kind of ion or different kinds of particles it lets through. So in this case, only potassium would go through a potassium leak channel. And in this case, as well as all the others, saturation can happen. If it builds up on one side, it will be saturated, say in the ECF, as it is trying to flow through into the ICF, but it's not needed, and so it can become saturated in the ECF. The second type of channel, we'll call this one number 1. I'll say this guy's number 2. Is a ligand gated channel. A ligand is it would attach to one of the sides here of the channel, and as a ligand comes in, we can just call this little triangle as the ligand. As it attaches here, it can open this channel. This channel, like the leak channel, is specific, so a specific ligand has to attach to the channel that allows the opening, and then when a ligand leaves, it would close the channel, and it would allow particles to flow in or out as needed of that particular channel. The third channel right here, number 3, is a voltage gated channel. So it looks just like a little gate. There's no extra parts to this. It opens and closes through charge, through voltage. So ions can be charged, like calcium has a +2 charge, potassium has a +1, chloride ions have a -1 charge. So as the ions build up inside or outside, a charge is created, and that can allow things to flow in or out. And then saturation will also occur. These voltage gated channels are specific to the ion that activates their gate to open or close. So in this case, we could call it sodium. So sodium's plus charge, as it builds up, it can open the gate, and as it leaves, it can close the gate with no charges, when the charge is no longer present. They also have can deal with saturation. As it builds up, it can create that charge and saturate, and then it'll open and let it through, and vice versa. And then this fourth channel is known as the mechanically gated channel, and they actually have springs along the membranes, And the way that they work is through compression and stretching of the membrane. As the membrane may stretch, it can open up this gate to come open, or if it were to compress, it could close that gate, and it would allow flow in and out of the ECF and ICF. It is also specific. All four of these channels have great specificity to what they let through. They're each specifically for whatever particle they were designed for, and saturation will affect all of these from the ECF or the ICF. The rate of flow for any of these channels is determined by either the number of particles, which could be a cause of saturation, or by the number of channels in the membrane. If there's more channels, you have a higher rate moving through. If there's less channels, then it's a lower rate that moves through. The next type of transport is a carrier-mediated diffusion. This is the phospholipid bilayer with one of those channels or one of those proteins in it that helps to transport particles. These proteins are very specific. They only allow certain things to bind to them. When something binds to them, it causes a shape change. It's very similar to a revolving door in a building. When it's open, it only opens to one side, and then it moves things through to the other side. So if we're in the ECF here and a particle comes in and it attaches here, then a shape change occurs. So it would become this shape where it closes off, and it would close off to both sides as it's moving through the carrier, and then it would finish by opening here and opening into the ICF, where we started in the ECF with that particle. There's no energy required in this. This is a totally facilitated diffusion with no types of energy. There is competition to get into these binding sites on these proteins, but there is no competition from other molecules because they are specific for each one. They have a specific rate that is set for them, and because of that, they do not, they do have saturation issues sometimes. If there is a buildup on one side for too many to move through, then saturation can happen on one side of the membrane layer. The next form of transport is a primary active transport. This type of transport does require ATP, so energy is used. Primary active transport is used when something needs to move against the concentration gradient, which means it needs to move from low concentration to a high concentration, because the typical gradient goes high to low. It is similar in a sense to how it works with the carrier-mediated diffusion, except this has a binding site for ATP to use. In primary active transport, it can move more than one ion or particle across the membrane, unlike in other cases. A common example of this is the sodium-potassium pump, or known as sodium-potassium ATP ACE. So I have the lipid bilayer drawn here with a primary active transport sodium-potassium pump here. So we'll call this the ECF and the ICF. So in primary active transport, you have three sodium that will go from inside the cell out. So you'll have 3 sodiums, and then you'll have 2 potassiums that are coming in. Potassium come in with ATP coming up and into this as a use. And what comes out is an ADP pi molecule. So this is a specific type of transport because it only pumps out, in this case, sodium and potassium, but it does do the more than one kind, like I mentioned earlier. And its rate is determined by how many of these pumps are across your membrane. The more pumps you have, the higher the rate of transportation that you can have. And just like in any other type of transport, saturation can occur. If there's more of one type of ion in the ECF, they can be built up and saturated in the ECF as it's trying to transport through to the ICF. And then in this type of transport, specifically in the ATPase pump, or the sodium potassium ATPase pump, there are two types. You can have E1 or E2. The E1 has the binding sites facing the ICF, so the intracellular fluid, and the E2 type has the binding sites facing outward to the ECF. But primary active transport, like I said, it does require energy. It's the first that requires energy that we've discussed so far. The last type of transport we'll talk about is secondary active transport. Like primary active transport, it helps to move things against their concentration gradients. But unlike primary active transport, ATP does not directly affect this process. So energy is created in gradients, and in this case, during primary active transport, there were 3 sodiums that were removed from the ICF, and that creates a gradient with enough energy to help us move other solutes. So sodium is typically what is used in secondary active transport. This protein will have 2 binding sites, one here for sodium and then one here for another solute. Once both of those things have bound into the protein, it will move something either in to the ICF, and that movement is known as a symport transportation, or if it drives something out from the ICF and back into the ECF, that is known as an antiport movement. The rate of any of this happening is dependent on the sodium potassium ATPase pump in primary active transport, because that's what gives us the sodium needed for secondary active transport. Much like every other type of transport, saturation can occur if there's a buildup. If things aren't happening fast enough, then saturation can occur in secondary active transport as well.